So in the previous video, we have described hexose and pentose molecules as linear structure. But this structure cannot explain a certain number of chemical and physical properties. So in this next video, we will show that in fact, pentose and hexose form cyclic structure. So let's, for example, talk first about the unexplained chemical property. Molecules that have a highly reducing aldehyde group are characterized by a very specific reaction called the Schiff reaction. In this reaction, a reagent called the Schiff reagent has been first discolored with sulfur dioxide. And when placed in the presence of an aldehyde, this Schiff reagent is recolored and turned pink again. Now, if a molecule of aldose is linear, we expect to see one free aldehyde group, and we expect that an aldose will recolor the Schiff reagent. In fact, what we observe is that an aldose fails to recolor the Schiff reagent, suggesting that the aldehyde group in an aldose is masked inside a cyclic structure. Now, what about unexplained physical properties? A linear molecule of D-glucose, for example, should exist in one form and have a unique set of physical properties. For example, a unique melting temperature and a unique degree of rotation of polarized light. In fact, when two distinct methods are used to chemically synthesize D-glucose, you obtain two forms that differ by their melting temperature, 146 degrees versus 150 degrees, and by the rotation of the polarized light, 112 degrees or 18 degrees. Furthermore, structural analysis show that hexose and pentose exist in cyclic structure. In fact, in solution, linear and cyclic structure are in equilibrium. However, linear structure represents only 0.02% of the molecules of sugar in this solution. So now, how can we form a cyclic structure? We have to go back to very basic chemistry. When an aldehyde reacts with an alcohol, you form an imiacetal. Similarly, when a ketone reacts with an alcohol, you get an imiketal. Now, in an aldose or a ketose, you have an aldehyde or a ketone group, and you also have alcohol functions, hydroxyl groups. Therefore, you can have an intramolecular hemiacetylation or hemiketylation, and that will lead to a cyclic structure. For example, we can start with a linear form of D-glucose and see how it can cyclize. We will start with a linear molecule of glucose and then bend the covalent bonds to bring the hydroxyl group carried by carbon 5 in close proximity to carbon 1 that carries the aldehyde group. Now, the formation of an hemiacetal is possible. And it can be formed in two different ways. The hydroxyl group on carbon 5 can attack the carbon 1 either from the front or from the back, leading to a new asymmetric carbon, a new chiral center, and as a consequence, two isomeric forms called alpha and beta. Let us consider for a few minutes some nomenclature. The carbon 1 that is involved in the cyclization is called the anomeric carbon in the cyclic structure. In the alpha isomer, the hydroxyl group carried by carbon 1 and carbon 6 are in opposite direction, which means that in our representation, the hydroxyl group carried by carbon 1 is below the plane formed by the ring structure. In the beta isomer, the hydroxyl group carried by carbon 1 and carbon 6 are in the same direction. That means in our representation of the beta isomer, the hydroxyl group is above the plane formed by the ring structure. Now we can go back to this difference in physical properties and can understand them now, because one method of synthesis leads to the alpha form of D-glucose, and the other method leads to the beta form of D-glucose. And these two isomers have slightly different physical properties.
Can ketose form a cyclic molecule?
Yes. Remember that they can form an hemiketal. So for example, in a pentose, the carbon 5, or the hydroxyl group carried by the carbon 5, can react with the carbon 2 that carries the ketone group to form a cyclic structure. In this case, the carbon 2 will be defined as the anomeric carbon. Now, you can form a large variety of cyclic structure. For example, you can form hexoaldose, pentoaldose, hexoketose, or pentoketose. So all of these molecules, all of these hexose and pentose, whether they are aldose or ketose, can form cyclic structure. Now, if we look at the structure of the ring, we can see that we have two different types. One that contains 6 atoms, 5 carbon and 1 oxygen. And this ring resembles the structure of a molecule called pyrane. And then we have a ring that contains 5 atoms, 4 carbon and 1 oxygen. And this structure resembles the structure of furane. So we can have a new nomenclature. For example, alpha D-glucose has a ring that resembles pyrane, and we can call it alpha D-glucopyranose. Or ribose, beta D ribose, has a structure that resembles a furan, and we can call it beta D ribofuranose. So why bother with a new nomenclature? In fact, this nomenclature is more informative because it takes into account the structure of the ring that is formed. And that is important when the same molecule can form two different ring structures. For example, glucose can form a stable alpha D-glucopyranose and a less stable alpha D-glucofuranose. Now we've described so far the ring structure as planar structure. It's not always the case. In fact, planar pyranose are not stable, and they adopt more stable conformation called chair conformations. We have in fact 2 of these conformations that can switch without breaking any covalent bond. There is also another rarer conformation called the boat conformation that we find in derivative of hexose that have very bulky groups attached to the carbons. So in cells, cyclical form of hexose and pentose are predominant. The cyclization results in the formation of an anomeric carbon and 2 isomeric forms for each hexose and pentose. In the next video, we will discover how monosaccharide assemble into disaccharide and complex carbohydrates.